A leaky blood-brain barrier is thought to play a role in different neurological and psychiatric disorders like brain fog, Alzheimer's disease, depression, schizophrenia, and more. However, the research in the field is still young, and scientists are yet to unravel the potential connections. When discussing issues with the blood-brain barrier, it's important to note that the problem doesn't always start in the brain. Diseases and other problems elsewhere in the body, like diabetes or gut inflammation, may trigger a leaky brain. Strokes cause a lack of glucose and oxygen, and this leads to a rise in potassium, the depletion of adenosine triphosphate, and the release of glutamate, and these all contribute to a blood-brain barrier disruption. Under stroke conditions, the tight junctions that help to prevent substances from entering the brain have compromised integrity, and this means the tight junctions are weaker and it increases blood-brain barrier permeability. After a stroke, reperfusion, which is the restoration of blood flow to the brain or the tissue, occurs, and this causes additional tissue damage and it increases the inflammation, and it also worsens the blood-brain barrier condition. In a study of human brain tissue from 5 cases of fatal strokes, MMP9 levels were higher in those affected areas, and higher MMP9 levels are a good indicator of increased blood-brain barrier permeability. After traumatic brain injury, the blood-brain barrier also becomes dysfunctional, and this leads to the leakage of proteins and negatively affects immune cells. Blood-brain barrier breakdown can last for several days to weeks after a head trauma. In some cases, the blood-brain barrier can be dysfunctional for years. Head trauma injures blood vessels in the brain, and this leads to impairment in brain blood flow, the blood-brain barrier permeability, and metabolic processes. It also reduces oxygen levels in the brain, and this further induces blood-brain barrier breakdown. Traumatic brain injuries also cause damage to the neurovascular unit. When the nerve cells die quickly, it causes another cycle of injury after the initial brain injury. After traumatic brain injury, excessive superoxide reacts with nitric oxide to form peroxynitrate. Peroxynitrate contributes to blood-brain barrier leakage. In traumatic brain injury-induced adult male rats, treatment to reduce the peroxynitrate also decreased the blood-brain barrier leakage. S-nitrosoglutathione helps to stop the blood-brain barrier dysfunction, and it provides protection to the nerve cells. There's a loss of tight junction molecules in brain tumor blood vessels. Tumors also affect cell formation, and they prevent the release of factors that are necessary for blood-brain barrier function. Brain tumors also cause fluid to leak into the brain. The open tight junction causes brain edema. In a study of 10 participants with brain tumors, brain scans showed an increase in blood-brain barrier permeability. Another study with mice who had brain tumors had similar results. The mice had increased blood-brain barrier permeability to sodium fluorescein, a marker of blood-brain barrier integrity. The blood-brain barrier breakdown is also associated with faster cancer growth. It's difficult to treat brain tumors because the blood-brain barrier prevents the transport of drugs into the brain, so scientists are currently studying new therapies that are able to cross this barrier. An increase in brain blood pressure can cause hypertensive encephalopathy. Symptoms include severe headaches, confusion, and impaired judgment and memory. Coma, convulsions, and other brain problems can occur afterwards. The breakdown of the blood-brain barrier during the increased brain blood pressure leads to reduced blood flow and to excess fluid in the brain. Normally, hypertension or high blood pressure activates pathways to maintain the blood-brain barrier. However, in animal models of hypertensive encephalopathy, hypertension overrides the blood-brain barrier. During the hypertensive encephalopathy, the blood-brain barrier dysfunction is widespread. It can cause brain edema, which is life-threatening, excess accumulation of fluid in the brain. Although there's methods to stop the brain edema, there's no treatment that specifically targets the blood-brain barrier. The obesity-induced inflammation can cause problems in the blood-brain barrier. Diet-induced obesity leads to an increase in pro-inflammatory cytokines, and this increases the blood-brain barrier permeability. Obesity also increases C-reactive protein levels. In rats, high doses of C-reactive proteins increase the blood-brain barrier permeability, and this impairs brain function. It mainly affects the tight junction function. High-fat diets and obesity cause blood vessel problems, damaging the blood-brain barrier integrity. In a study of male rats, in comparison to mice fed a healthy diet, a high-fat diet increased the blood-brain barrier permeability, and moreover the results of the study suggest vitamin D supplementation helps to protect the blood-brain barrier. Although it doesn't seem to have any effect on the control group, vitamin D treatment significantly improved the blood-brain barrier integrity in rats who were fed a high-fat diet. The rats fed a high-fat diet but didn't receive the vitamin D supplementation had higher blood-brain barrier permeability. Blood-brain barrier permeability is increased in diabetic patients and animals. Diabetes can result in the disruption of the blood-brain barrier. Elevated sugar levels increase the oxidative stress in the brain and the pericytes, and this kills the pericytes by apoptosis, and it disrupts the blood-brain barrier. By blocking glycolysis, or the breakdown of glucose, the blood-brain barrier pericytes are protected. During diabetes mellitus, reactive oxygen species accumulate at the blood-brain barrier, and this causes oxidative damage, further damaging the blood-brain barrier and increasing its permeability. Researchers tested the blood-brain barrier selectivity in diabetes-induced rats, and results indicate that diabetes caused the loss of tight junction proteins, and it increased MMP9 activity, which leads to an increase in blood-brain barrier permeability. Leaky gut happens when the permeability of the intestine increases, and this can also contribute to a leaky brain. The blood-brain barrier helps to exchange information between the brain and the gut. The blood-brain barrier transports peptides and proteins from the brain to the gut and vice versa. Stomach hormones affect the blood-brain barrier function. Gut peptides can also inhibit or increase the release of cytokines from brain cells. Changes in the gut also influence the permeability of the blood-brain barrier. The blood-brain barrier of mice is vulnerable to changes in the gut microbiota. In germ-free mice with no gut bacteria at all, exposure to gut microbiota from pathogen-free mice reduced the blood-brain barrier permeability. In addition, leaky gut may contribute to the onset of inflammation and diabetes, which also contributes to blood-brain barrier damage. HIV infection and viral proteins damage the blood-brain barrier's integrity. This also contributes to the progression of HIV-associated brain disorders. During HIV infection, the virus infects the CD4 T cells and the monocytes, and this then crosses the blood-brain barrier and spreads the infection. Because the blood-brain barrier doesn't allow for the easy transfer of foreign molecules, it's hard to treat HIV. In HIV-infected tissue, the blood-brain barrier had high permeability, cell death, and an abnormal interaction with blood vessels. Following HIV invasion, pro-inflammatory cytokine levels also increased, and this can contribute to blood-brain barrier permeability. The HIV GP120 protein causes an increase in reactive oxygen species, and the increase in the oxidative damage also contributes to the blood-brain barrier dysfunction. Although combination antiretroviral therapy helps to decrease the viral load in the body, it can't lower the virus level in the brain. Nanocarriers have the potential to cross the blood-brain barrier and to reduce HIV virus levels. More studies are needed to be performed before an effective therapy is found. Bacterial meningitis infection disrupts the blood-brain barrier. During meningitis, TNF-alpha damages the blood vessels, causing an increase in blood-brain barrier permeability. There's many ways that bacteria disturb the blood-brain barrier. The ways include disrupting the tight junction through the toxic effects, including inflammation or interfering with the junction formation. In one study, 48 patients with bacterial meningitis had significantly higher levels of TNF-alpha when compared to 66 controls. The higher levels of the TNF-alpha correlate with the higher albumin level in the blood, and its pro-inflammatory effect contributed to the blood-brain barrier damage. Lyme disease is an infectious disease. It's a bacterial infection which is transmitted to humans by a bite from infected ticks. The B. burgdorferi bacteria invade the immune system, and they spread to the brain and the nervous system by crossing the blood-brain barrier. Spinal fluid sample from Lyme disease patients showed higher levels of blood proteins, fibrinogen and immunoglobulin compared to healthy subjects. This is evidence that the blood-brain barrier's integrity is compromised during Lyme disease. Researchers used to think that the bacteria induced plasminogen activators and MMP production. These enzymes degrade tight junction proteins that help with the blood-brain barrier selectivity. This increased blood-brain barrier permeability allows for the bacteria to cross it. However, in a study of human cell culture, there was no indication of tight junction protein dysfunction. The exact mechanism by which the bacteria lowers the blood-brain barrier permeability is still unknown. Alcohol abuse causes blood-brain barrier dysfunction. The increase in the blood-brain barrier permeability leads to brain damage. After consumption, alcohol readily crosses the blood-brain barrier. The alcohol damages the tight junctions in the gut, and it allows for the LPS lipopolysaccharides, a bacterial toxin, to leak into the bloodstream, and this eventually leads to the release of pro-inflammatory cytokines in the body that cross the blood-brain barrier. Lipopolysaccharides are gram-negative bacteria cell walls. In one study, researchers concluded that one episode of binge drinking doesn't cause brain damage. However, repeated cycles of alcohol consumption and withdrawal cause severe blood-brain barrier dysfunction and brain damage. In rats, alcohol consumption caused the blood-brain barrier to be more susceptible to damage from lipopolysaccharide toxin injection. It prolonged the inflammatory response, but it suppressed programmed cell death in the nerve cells. Chronic alcohol drinking makes you more susceptible to infection and inflammation-related diseases by letting toxins pass through the blood-brain barrier. The liver releases the breakdown products of injured liver cells into the bloodstream during acute liver failure. This includes pro-inflammatory cytokines like TNF-alpha, interleukin-1, interleukin-6, and ammonia. These cytokines cause brain inflammation. The inflammation causes the blood-brain barrier to become more permeable. Blood-brain barrier dysfunction causes fluid to accumulate in the brain. In one study of post-mortem acute liver failure patients, 80% showed evidence of fluid in their brain. In acute liver failure patients, the injured liver releases MMP9 into the bloodstream. The circulating MMP9 damages the tight junctions at the blood-brain barrier. This increases the blood-brain barrier permeability, and it allows brain damage to occur. It's possible for the blood-brain barrier permeability to return to normal levels if the patients quickly receive a liver transplant. However, there's still a possibility that the transplant will occur too late while the brain injury still progresses. For information on some natural ways to help to repair a leaky blood-brain barrier, check out my next video. And for more information on herbs, supplements, and natural treatment plans, check out my website.